Okay, here we go with our DNA notes. Okay, so as an overview, we are going to talk about what DNA is, DNA structure discovery, the actual DNA structure, and then DNA replication. Okay, so the first thing we need to talk about is what is DNA? DNA is the set of instructions for all the things a cell can do. So making proteins, making membranes, just doing everyday life, replicating, anything a cell needs to do, DNA is the instructions. Okay. DNA has three main functions. The first one is to store information, the second is to copy information, and then the third is to transmit it. Each one of those is just as important as the next one. So first of all, storing information that sits in the nucleus with the DNA. Copying information would be when the cell is copied and then the DNA is copied along with it. And then to transmit information, that means to take the message that's inside the DNA and turn it into something that's usable. Okay? And DNA, the word DNA comes from deoxyribonucleic acid. So the deoxyribonucleic acid. Deoxyribose is a sugar, and then nucleic acid refers to one of the four compounds that can be found in our cells. So nucleic acids, proteins, carbohydrates, and fats. Three of those we can take in through our diet. And of course DNA we have to make. Okay. So the discovery of DNA has a pretty crazy history. First of all, a couple scientists that aren't mentioned on here, like Avery and Hershey and Chase, they first discovered that it was DNA that was doing the inheriting, that was the control of the cell versus protein, through some experiments with mice and bacteria and things like that we won't cover now. But if you're interested, you can ask me and I will talk to you a little bit more about that. Okay, a couple other people that were very important in the discovery of the DNA structure. One was Erwin Chargaff, who discovered the base pairing rules. Okay? And so here is a key answer for the end. The key answer is the base pairing rules are the set of rules that govern how DNA structure comes together. So we say that always adenine and thymine pair together and guanine and cytosine pair together. Okay, then there was Rosalind Franklin. Rosalind Franklin was in the top of her field, especially for a woman in the 1950s. And she worked at taking X-ray pictures of DNA. She later ended up getting cancer from all of the radiation from the pictures. But as you can see in this picture over here, you can see the X-like shape of the DNA leading to the thought that it was a double helix. Okay. And then finally, Watson and Crick. They're the ones that actually built the model of DNA. So they didn't discover DNA, but they discovered its structure and built the model. They first made a 3-stranded DNA molecule, but that didn't really work. And then they were kind of laughed off the scene and told, hey, you need to go back and not ever do this again. But they kept going, and they came up with the 2-sided molecule or 2-stranded molecule that we now know. So with the help of Franklin's X-rays and Chargaff's data with the A equals T and G equals C, they were able to win a Nobel Prize for discovering the structure of DNA. Okay, so let's get a little bit more into that structure. Okay, so first of all, the structure is made up of smaller things called nucleotides. Okay, so each little piece of the DNA is called a nucleotide, and there's a bunch of nucleotides that are strung together to equal the whole molecule of DNA. There are 3 parts to a nucleotide: the phosphate, the nitrogen-containing bases, which are adenine, thymine, guanine, and cytosine, and deoxyribose, which is the sugar. Okay, and they're pictured down here. Okay. A little bit more about the structure. So again, those 4 nitrogen bases are adenine or A, thymine or T, guanine or G, and cytosine or C. Okay. So here is the base, here is the sugar, deoxyribose, and the phosphate group. And each one of those all together makes up a nucleotide. Okay. The sugars and the phosphates right here make up the outsides of the DNA molecule. So if we were comparing it to the ladder, those would be the sides of the ladder. And then the bases are on the insides. Okay, and I'll circle this one in green too. So that's the outside, phosphate and sugar. And you see how they alternate, phosphate, sugar, phosphate, sugar. Okay, and then the bases are on the inside. Those bases are held together by these important hydrogen bonds that form between the molecules inside of, or the atoms, excuse me, inside of the DNA. Okay, and then we already went over those base pairing rules that A pairs with T and G pairs with C. Okay. The structure of DNA is that it is a double helix shape with two sides. So side 1, side 2. Okay, so we could actually divide it if we cut those hydrogen bonds there. So there's the two sides. Okay. Another key answer is that DNA is said to be complementary, which means that one side goes with the other. So to complement something means they go together. Okay, so now let's get a little bit more into those base pairing rules. We know the amounts of adenine and thymine are equal. Shargoff told us that. And the amounts of guanine and cytosine are also equal. Now when I say equal, I don't really mean truly equal, but they're really close, like within a few tenths of a percentage for each. So we're going to assume that they are equal. Okay. Now, if we take a normal cell, we know that there can only be 100% of DNA in the cell. There can't be more than that because the 100% is all there is. So with that in mind, given any percentage of any base, we should be able to calculate the other three. Okay? So here's our example here. If we are given guanine is equal to 10%, we also know that cytosine is equal to 10% because guanine and cytosine are equal. Okay, so if we get 10 plus 10, that equals 20%. Well, 100 minus 20 leaves us 80% for the other two bases, A and T. And we know A and T are also equal. Okay, so if we take 80% and divide by 2, we get 40% for adenine and 40% for thymine. We'll have a couple more practice problems with this, and I'm happy to explain a little bit further individually to anybody that's really unsure or doesn't really get this. Okay, so a big thing to remember here is that there can only be 100%, and that adenine and thymine are equal, and guanine and cytosine are equal. Okay. So here's another picture of the DNA, a little bit more of what it looks like. So here is more of the cartoon image. This is more of a molecular structure, what that would look like. This would be called, a ball and stick model. If we actually looked at all of the carbons and nitrogens and hydrogens, it would get really complicated. So we're not going to do. Okay, so now the next topic we need to talk about is DNA replication. So we all know that DNA has to replicate before cell division because we want each new cell to have a full set of instructions. We talked about during the cell cycle notes the problems that could happen if DNA wasn't copied, and so we could have one cell without any DNA, we could have a cell with too much DNA, and neither of those situations would be good. Okay, so we're going to talk a little bit about how that process happens. So replication is just simply making a copy of the DNA, and we know that this happens during interphase, before the cell even goes into mitosis or cytokinesis. So DNA replication happens at this thing called a replication fork. A replication fork is where two sides of the DNA are separated. Okay, that's very important that we have to separate the DNA because we have to break those hydrogen bonds. Okay. The cool thing about DNA, as another key question, or key answer, excuse me, is that both sides, this side and this side of the DNA, are being replicated or copied at the same time. Okay. When the DNA is being replicated and copied, there's two strands. You can see here there's a 3, it says 3', and then down here is the 5'. So the lagging strand takes the 5' and makes a 3', and the leading strand, which is here, takes the 3' and makes a 5'. You don't need to focus too much on the 3' and 5', but I just thought that was interesting how both sides are copied at the same time and they're going in separate directions to avoid confusion. Okay, so let's learn a little bit more about the actual process. So the first part of DNA replication is that DNA helicase, DNA helicase, which is an enzyme, has to unwind the DNA or unzip the DNA, if you've ever heard of that, which basically just means it's breaking the hydrogen bonds. Okay? We can't start replicating until we've pulled those base pairs apart and they like each other, so they're really stuck together. Okay. After that section of the DNA is pulled apart by the helicase, another enzyme, polymerase, comes in and it's going to add a new nucleotide to each side. Remember what the 3 parts of a nucleotide are: phosphate, sugar, and base, and it has to assemble all of those. Another key answer, anytime you see ASE at the end of the word, it's usually an indication that it is an enzyme. Enzymes are things that help to speed up reactions. Otherwise, they would go really, really super slow. Okay? So step 1 was helicase unwinds the DNA. Step 2 is polymerase comes in and nucleotides. And then we get our final result. We have 2 identical DNA molecules. Hopefully no mistakes were made, and if mistakes were made, they were caught and fixed, which DNA has checkpoints that help it to do that. Okay? Each molecule has 1 old strand and 1 new strand or side. Okay? So we can see in this picture, pointing to the old strand of this one and the new. So purple is old, pink is new. And then over here, purple is old and pink is new. So this will eventually unwind all the way up to the very end of the DNA and replicate all the way through, and we'd have 2 separate molecules that each have an old strand and a new strand. Here's the final key answer, is that this is called semi-conservative. It's called semi-conservative because we're keeping half of the old DNA in each one. So it's not completely conserved, but we're half conserving it. Okay, so hopefully you've learned some things about DNA replication, and we're about to go through those key questions now. Okay, finally we get to the end to those key questions. So all throughout the presentation, I've been giving you key answers, and now here are the key questions. What I'd like you to do with these is just write the answers. So you don't have to worry about writing the questions unless, of course, you would like to at the end of your notes. And then, so you'd write number 1, and then give me the answer to what are the base pairing rules. So hopefully if you've been listening while you've been taking notes, you've heard the answers to these 5 questions. Again, don't forget to come up and show me your notes to get your points, and I also will be looking for the answers to these 5 questions. Have a great day, and I will see you in class.